So did you have a problem?
Welcome. It's terrific to see you all here. Thank you for finding your way through the maze of the Kennedy School. It feels like some kind of experiment. You know, there was sort of something at the end of a long, complicated tunnel, and some of the mice made it, and some of the mice didn't make it. So I'm glad you're in this group of mice, all your mouse models, George. So this is the first lecture in this year's public lecture series for the Edmund J. Stafford Center for Ethics. Many of you have been part of our community for some time, so you know that at the center we seek to strengthen teaching and research about pressing ethical issues, to foster sound norms of ethical reasoning and civic discussion, and to share the work of our community in the public interest. Few issues could be more important than the theme that the center is pursuing in its fellowship program this year, the ethics of technological and biomedical innovation. We're all fully aware that the world at this point has many powerful hubs advancing technology, but at the same time, few addressing the urgent human dilemmas raised by technology. And by tech, I'm thinking both about computer and biomedical forms of innovation. So a range of dilemmas brings up questions of ethics continuously, questions of governance, corporate decision-making, and the changing shape of what it means to be human. And Harvard is really a natural place to launch a significant initiative in tech and human values. So Harvard, here we have just incredible resources. The world's leading liberal arts college. We're a research university. We have the world's top professional schools, for example, the Harvard School of Medicine. And we're doing all this intellectual work across these areas in a city with cutting-edge technology companies in both the computer science and biomedical domains. So there's a real opportunity for us to be the people who take the dilemmas emerging from our current moment the most seriously. There's a real opportunity for us here at this place to be leaders in trying to come up with solutions to the dilemmas that emerge from technology. So the Stafford Center has partnered recently with the Berkman Klein Center for Internet and Society to really try to spearhead an initiative in tech and human values. And the goal of this initiative will be to transform how technologists, scientists, public servants, enterprise leaders, and professionals learn to see and respond to ethical and governance dilemmas provoked by technology. The initiative will accelerate the discovery of solutions to high-priority dilemmas, from disinformation to algorithmic bias to gene editing, we'll talk about that tonight, and policy based on polygenic science. Our public lecture series presents people committed to modeling insightful identification of the ethical stakes of personal, professional, and public affairs. Our lecturers inevitably model sound norms of ethical reasoning and help us all have productive civic discussion. With our lectures, we hope to provide an intellectually stimulating and supportive environment for our fellowship cohorts and student, faculty, and staff affiliates. I can think of no better speaker to launch our series this year than George Daley, Dean of Harvard Medical School. He's a world-renowned physician scientist and educator. His achievements span basic science, translational research, and clinical medicine. He's at the forefront of stem cell science and cancer biology, and his discoveries have twice been cited in Science's top 10 breakthroughs of the year. I could go on about his work in the lab, mentoring younger scientists. He made important contributions to the university's Inclusion and Belonging Task Force by helping us think more productively about mentoring. But then perhaps the most important detail in his biography, not though on his CV, is the fact that as an undergraduate at Harvard College, he studied with John Rawls. So he has the official credentials to be here tonight, kicking off our year of discussion and reflection, action, and policy development with a lecture titled Ethical and Social Challenges of Emerging Biotechnologies.
Okay, thank you very much, Danielle. I have to correct a little bit of the public record. I'm not an ethicist. I am a physician scientist, but true, for a year and a half, I did claim my undergraduate concentration in philosophy. And that did give me a semester tutorial with Rawls, where we spent the entire semester reading the groundwork. So a whole semester spent on 60 pages of something that I think has certainly been always in the back of my mind, the notion of moral reasoning in the face of all of the challenges that we are confronted by as practicing physicians. And then when my scientific career started to evolve, I found myself drawn into the public debate around stem cells, in part because I was one of the few scientists working in the area of embryonic stem cells who was also a physician and could put the potential of stem cells into a compelling medical context. And I find that over the last 20-some years, again and again, I am brought back into the discussions at the interface of medicine, technology, and bioethics, and I found it a deeply, deeply rewarding and challenging enterprise. I'm going to talk today about genome editing primarily, but I will also raise a number of other emerging technologies as they challenge us to think about the responsibilities of physicians and scientists to invite scrutiny of their work, to conduct their work under either institutional review or broad professional norms. And I'll talk a little bit about our latest attempts as the members of the International Society for Stem Cell Research to provide a comprehensive overview and a framework for doing this work in a responsible manner. I imagine virtually all of you read this book at some point in a high school literature course, which I did, but then recently picked it up again. And Brave New World remarkably was published in 1932, and yet it was remarkably prescient because it anticipated in vitro fertilization. But it also described a dystopian view of the future where reproduction became highly mechanized, where embryos were cultured in hatcheries, where they were genetically modified and conditioned, and individuals were pre-sorted into castes. And I think it was certainly a cautionary tale, but at the time, the Brave New World required in vitro fertilization and genome editing. The reality is that this is no longer science fiction of the future. These technologies are now broadly facile. So I want to talk about some of the ways that the scientific community has attempted to come together to regulate the practice of their own science. And it would start with the era of recombinant DNA. Paul Berg was recognized with the Nobel Prize for being the first to create a novel hybrid splice of DNA from distinct organisms. His experiment began with the SV40 virus, a monkey tumor virus, and cutting and splicing that DNA with the virus of the lambda phage, a small virus that infects bacteria, that you will hear more about. That experiment created the first hybrid DNA molecule between two organisms. The next phase that he envisioned, hi, how are you doing, Allison? The next experiment that he envisioned was to propagate this hybrid DNA in a bacterial cell so it would be amplified. Now, the bacterial cell that has been the workhorse of biotechnology is Escherichia coli, which is a denizen of our gut. And to Paul's credit, he recognized the potential risk of propagating a tumor virus in a denizen of our own gut and stopped the experiment, and then called attention among other scientists to the potential risks of recombinant DNA. In a very famous meeting, he gathered about 150 of his colleagues in Asilomar, California, many of the leading figures in molecular biology, Jim Watson, Sidney Brenner, my thesis advisor, David Baltimore, Maxine Singer, and the like. And the objective there was to try to assess the scientific foundations of this new technology with regards to safety. And indeed, they very productively were able to project what they thought was a safe set of containment principles, which are now widely used around the world and have resulted in an extraordinary safety record for the recombinant DNA revolution. And it was, in many ways, an establishment of a principle of self-regulation by scientists. Now, a subtext of the discussions was the recognition that being able to splice DNA suggested that one day we might be able to actually alter our own DNA, our own heredity. And that, of course, created a set of quandaries, but the deep analysis was left for the future, because it was believed at that time, just as it was believed in 1932 when Huxley wrote Brave New World, that this technology was so far in the future that it wasn't necessary to exert a lot of energy on it. Well, the future is now. Now, since 1975, there has been a dramatic application of gene splicing and recombinant DNA in modern medicine. Within just 8 years of the first publication of recombinant methodology was the first production of human insulin. And following that were many, many proteins and biologics that really transformed medicine. Also, we can now use viruses safely to transfer to cells normal copies of genes and treat individuals who are born with inborn errors of genetics. And in fact, for the 9 years before I was dean, I was running the bone marrow transplant program at the Children's and the Farber, and we were starting to see more and more patients on our service that we were literally curing for a variety of diseases. Immune deficiency, hemoglobin disorders, and in other clinics, there's great promise for diseases like hemophilia, blindness, and the like. All the fruits of recombinant DNA technology. But there are limitations, and scientists are never satisfied, thankfully, keeps us in business. Gene therapy works in a limited range of tissues. We know from early experiments, and every experiment is tainted by worry about the risk, but in the first cases where children were treated for immune deficiency, the virus hopped into a region of the target cells that activated cancer genes, and kids, some of the early kids came down with leukemias. Also, we know that not all genes can be corrected by simple replacement. Their modulation and their regulation is essential to a cure, and viruses don't provide a natural form. So this has driven the need for more precision and the technology around genome editing. So one of the great breakthroughs, let's say, of the 90s and the first decade of this century were the creation of proteins that could recognize specific sequences of DNA. These were proteins that were components of zinc fingers or tailings, and they were fused to enzymes that could cut DNA. So what these allowed us to do was to create a targeted sequence-specific cut in DNA. And we know that when a cell suffers a double-strand break of the DNA, it attempts to heal that break, and in so doing, will rearrange and mutate the DNA so as to lose the function of that region of DNA. Now, the problem with this is that every single sequence that's going to be targeted requires a new protein, and the design and optimization of that protein requires years of effort. So it's a very cumbersome, a very challenging technology, and yet this technology is in fact in the clinic to inactivate the gene that is the co-receptor for HIV, and patients are being treated in an attempt to induce resistance to HIV with this technology. Now, why did things change? this is one of the great stories in all of the history of science, because it's a testimony to the remarkable ability of scientists who are curious and set out to solve a problem, to make discoveries whose implications no one could see. I'll talk about CRISPR, and it's really the work of Rodolphe Barringau and Phil Barbath. They are food scientists working for the European company Danone. And Danone was recognizing that some of their production facilities for yogurt had cultures that were robust and productive, and others seemed to crash and deteriorate. And so these food scientists were assigned the task of figuring out why yogurt cultures were failing. And what they learned was that they essentially went to the facilities and captured strains and did comprehensive sequencing of DNA. And they compared the DNA sequences of the robust cultures against those that were susceptible to death. And what they found was something quite fascinating. Those cultures that were dying were subject to infection by a lambda phage virus, that same virus that Paul Berg used to make the first spliced piece of DNA. And what they learned was that those strains of bacteria, fermenting bacteria, that were robust were immune to the bacteriophage that were destroying other cultures. They had acquired resistance by integrating copies of the virus into their own genomes, into their own DNA, and then they would express, sort of mimic copies of these infecting viruses, and together with a protein that would recognize the homology of the original virus with a new invader, the protein would recognize that and cut it. So it was a way of making a sequence-directed DNA nuclease, a cutting machinery that rendered the cell resistant. Now this is known as CRISPR, which actually stands for clustered radially interspersed short palindromic repeats. CRISPR rolls off the tongue a lot more easily. So they recognized that this was an adaptive immune system in bacteria, and it's a fabulous contribution to basic science. These systems now are widely known across almost all bacterial species. Now, the real transformation, though, and I'm going to argue, but I only get one vote. I borrowed from Luciano Marafini from Rockefeller some very, very nice visuals. So I'm going to show you, I'm going to take you through the way this actually works. So here's the phage electron micrograph showing these little phage all decorating a bacterial cell. And here's the components that are the Cas9 CRISPR components. So this is an RNA, which is a nucleic acid that can be designed against any target DNA sequence. And what it will do is it will scan any DNA sequence and find its match. DNA, RNA hybridization. And when it finds its match, it will cut. And that is the essence of CRISPR. That is the essence of CRISPR. Now, what is so brilliant about this is that 3 scientists looking at the mechanism by which this nuclease RNA system worked to cut DNA recognized that they could splice together what were 2 independent RNA components of this system into one molecule that would be loaded onto a protein and directed at a specific sequence in DNA. This is now a very facile system. Instead of taking years to design sequence specificity, any one of the scientists in my lab can go online and in an afternoon design thousands of these, and 2 days later be able to perform an experiment. So what took years before now takes hours to days. And what one can do is you either repair these cuts in DNA by a process called non-homologous end joining, and that leads to the inactivation of a gene, or alternatively, you can provide a template piece of DNA which will repair the cut that you've made, and that will lead to gene replacement. So now we have a remarkably facile strategy for editing genomes. Now, instead of the technology being decades in the future, the technology is practiced by virtually every molecular biology laboratory in the world. And I'm gonna guess that these 3 scientists are going to be recognized with the Nobel Prize, if not this year, perhaps next year. So this is already in the clinic. If you can imagine, this paper that was published by these 3 individuals in 2 different publications came out in 2012. And here we are in 2019, and the first patients are being treated. There's an effort by the National Institutes of Health, the Cure Sickle Cell Initiative, and I won't belabor the details of the molecular biology, but sickle cell anemia is an abnormal form of the protein that makes red blood cells red and carries oxygen. And there is another protein within the cell that can be reactivated to protect the cell against sickling, and CRISPR is the strategy for that reactivation. And I think this is an exciting, exciting development, and it shows how rapidly now we can move from fundamental science to curing patients. So there is no controversy whatsoever about the somatic or use of this gene editing technology to repair damaged body tissues. The question is, are we now at the point where we have a technology that allows us, in fact, to edit our own heredity? So I told you that the transformative paper was published in 2012. And within a year, scientists at MIT, fostered by transformative breakthroughs of Feng Zhang and George Church at MIT and at Harvard, were able to engineer the genomes of mice, and just 2 years later, were able to engineer the genomes of monkeys. So around 2014, 2015, a number of us in the scientific community started to speak about the inevitability that this technology would be applied to human embryos. And it was a remarkable confluence of experiences, but I was called for a conference call with some representatives from the Public Policy Committee of the International Society for Stem Cell Research, where I was engaged, and they were talking about rumors of papers circulating reporting the genome editing of human embryos. And literally the day of that conference call, I received in my inbox the request to review 2 different papers reporting the genome editing of human embryos. And indeed, within a few months, the first of those papers was published. I should say, I actually rejected the paper for scientific, not any other grounds, and it ended up coming out in a very, very essentially unknown journal, Protein and Cell. But it was an interesting report because the first report involved what are called tripronuclear human embryos. So typically, egg, sperm come together, and a single sperm will fertilize the egg. But occasionally, you'll get polyspermy, and you'll get 2 sperm in the egg, and that leads to a non-viable embryo. And this initial experiment was done with these putative non-viable tripronuclear embryos. I think in an attempt to reduce the ethical furor that might erupt in the publication of this work. But needless to say, there was a tremendous amount of consternation about this experiment. And there was hue and cry. And so Jennifer Doudna, one of the discoverers and developers of CRISPR, together with Paul Berg, David Baltimore, convened a meeting of scientists, and I was one of those invited. And we met in a very, very nice place in Napa Valley, which for those of you who know me, I'm a wine lover, and that was an extra inducement to participate in this meeting. But we seriously came together and said, there is an inevitability here. Someone is going to try to use this to birth a baby. And we all knew that the safety of this had not been established. And we were deeply concerned. And indeed, we called for an international forum to discuss this issue. And the United States National Academies, working together with the Royal Society in London and the Chinese Academy of Sciences, convened an international summit in Washington, D.C. in December of 2015. And David Baltimore chaired the organizing committee on which I served. And in his introductory comments, he said, "We are here as part of a historical process that dates from Darwin and Mendel. We're taking a heavy responsibility for our society because we understand we could be on the cusp of a new era where we are able to alter human heredity. And now we must face the questions, how, if at all, do we as a society want to use this capacity?" Well, the organizing committee, after hearing from a very diverse array of stakeholders, speaking from different religious perspectives, different bioethical perspectives, patient advocacy perspectives, concluded that embryo editing, regardless of what you thought of its propriety or ethics, was irresponsible because it was unsafe. And it called for further active discussion of both the scientific efficacy and safety, but also deeper social engagement to think about what, if any, would be the circumstances by which one might practice this. And indeed, taking this to heart, the U.S. National Academies convened a study group, and they published a very comprehensive and quite engaging analysis of the various issues. And they concluded, quite interestingly, that germline editing research trials, and germline editing meaning those edits primarily of human embryos, but they could also be of human gametes. But those edits that would not only find their way into every cell of the person born, but would be passed on to the next generation through the germline, that these trials might be permitted, but only after more research to understand the appropriate risk-benefit standards for authorizing the clinical trials. And another group, the Nuffield Council on Bioethics, produced about a year later a slightly even more permissive stance, saying we can indeed envisage circumstances in which heritable genome editing interventions should be permitted. And I would say in the period between 2015 and 2018, there were over 60 groups internationally who came together and wrote a policy piece or a report of inquiry establishing a range of positions on the permissibility of genome editing. So we had in 2015 called for another international forum at some time in the future. And we had imagined that that forum would continue to be sponsored by the US National Academies, the UK Royal Society, and the Chinese Academy of Sciences. But as we were working towards the planning of the meeting in Beijing, we were told by the organizers that we would not be allowed to have a meeting that had any more than 50 people, and we would not be allowed to have press and have it be transparent and open to the public. This to us was essentially a tacit rejection of our being able to hold the meeting in Beijing. And we realigned with the Hong Kong Academy of Sciences and ultimately held the meeting in Hong Kong. And I remember that very exhausting flight into Hong Kong and landing and having my phone turn on and all of a sudden having about 20 texts and emails because we had gotten advance notice that there would be the reporting of the actual birth of genome-edited babies. Jiankui He, a Chinese scientist who had pursued postdoctoral training here in the United States, had in fact pursued gene editing and reported it to probably the greatest media frenzy I have ever witnessed. When he came to the stage, there was this phalanx of hundreds of reporters, and the shutters were going off at such an amazing pace that Robin Lovell-Badge, who was chairing the session, tried to shut them down, tried to say, no more cameras, how many pictures do you need? And they started screaming out, freedom of the press, and all that. Anyway, the cameras finally did quiet enough that He was able to give his talk. Immediately afterwards, David Baltimore stood up, and in a remarkably direct confrontation, he condemned the work, called it irresponsible, and called for an international inquiry. Indeed, we actually don't know the disposition of the scientists. We don't know the disposition of the inquiry. And I'm certainly deeply concerned that we may not ever know actually how much institutional complicity there was in this. But we do know, as captured by a very pithy report in The Atlantic, that this was a failure on many accounts, a failure based on the international norms that had been established through those studies. First of all, the genome, or the gene that was targeted, was this co-receptor for the HIV, the AIDS virus. And it was not clear, as I'll show you, that that would have been the first choice. If an international scientific committee had come together to think about the risks and benefits, this would not have risen to the top. It was clear from even the data presented that it was a failed experiment. The editing did not replicate the naturally occurring gene deletion that renders about 1% of Caucasian populations resistant to HIV. So what edits were done in these children had an unclear impact on their ultimate susceptibility to HIV. The issue of informed consent, the scientists claimed that indeed there had been informed consent and there had been institutional review. But in translations of the informed consent documents, it was clear that they did not meet even a close standard of an international form. This was done in a clear effort to suppress any knowledge, and thus there was a lack of transparency, and there had been no public engagement. There had been no consensus. There had been no sense that it was time and this was permissible. Now, there are counter opinions, and interestingly, our own Noah Feldman wrote a rather provocative piece saying that the fear of designer babies is overblown and the disease-fighting benefits too great to ignore, and imagined that the umbrage and outrage would fade fast. Indeed, we now know from broad public surveys that if you ask about the applications for disease, there's about a 70% frequency of endorsing the use of gene editing technologies. But if one talks about modifying disease risk or enhancing human potential, the support plummets quite fast. Well, as a physician, one of the few physicians on the organizing committee in 2015, 2018, I was given the unenviable task of putting forth compelling medical rationale for genome editing. And this is a framework that I developed that essentially looks at the spectrum between something we might greenlight and something we might prohibit. And as has been supported by public surveys, using this technology to prevent devastating diseases, the genetics of which we deeply understand and we don't think would cause any significant unknown complications were we to revert the disease allele to normal. However, if it's just modifying disease risk, it's a little bit more uncertain. We could modify HIV resistance, could modify heart disease. There are even folks, Stephen Hawking, before he passed away, was arguing that indeed it would be imperative in the future that humanity would take control of its heredity in order to survive in a changing climate. And indeed, if we face the likes of global pandemics, there might be an increased acceptance of the possibility of building resistance into our own genomes. Far more concerning, however, are the range of gene changes that we would consider enhancements. Believe it or not, there is a gene that correlates with body odor, and there are many who might think that wouldn't be a bad thing to eradicate from the gene pool. But I think what's driving a lot of this is the fascination with the notion that there are genetic underpinnings to learning memory. I think it's fair to say that part of the motivation from Jiankui, the Chinese scientist, to use the HIV CCR as a target was that it had been linked to enhanced learning in memory in an experiment done at UCLA. I would argue that most of the traits that we might search for in the sense of enhancing our kids, creativity, musicality, bravery, intelligence, whatever, these are complex polygenic traits that clearly involving gene-environment interactions that certainly we have no ability to control today. But it really raises the question of how do we as a society decide whether any of these are permissible or impermissible? Now, always in the context of thinking about risk-benefit, one has to weigh the alternatives. And as I said, for conditions like sickle cell anemia and cystic fibrosis, there's now a growing number of very, very disease-altering, if not curative types of interventions. So there is a strategy called pre-implantation genetic testing or pre-implantation genetic diagnosis, where couples can perform in vitro fertilization and then genetic testing on those embryos and select those embryos that are disease-free. Now, unfortunately, there are rare instances where there are no disease-free embryos. There are certain conditions, such as Huntington's, which are autosomal dominant. You only need to inherit one copy of the bad gene. But in some clusters, particularly in Venezuela, where Huntington's disease is highly endemic, there are individuals who are homozygous for the Huntington's allele. They have two bad copies. They have no possibility of having a child without the disease. And then, of course, there are conditions. Let's say if two individuals with sickle cell anemia decided to have a child, there would be no healthy embryos. So the only opportunity there for a genetically related healthy offspring would be gene editing. Now, it has been argued by some that these instances are so rare that they don't warrant our developing the technology, and that pre-implantation genetic diagnosis is enough. But if one actually looks into the data, as was done by Stefan and colleagues, looking at the comprehensive, almost 20-year experience with pre-implantation genetic testing at the Necker Hospital in Paris, what they found was that the probability of success was quite low, and that there was a failure rate of almost 3/4. And they speculated that this might be a permissible attempt to allow families to have genetically normal children. So in the aftermath of the announcement of the Chinese twins and the 2018 summit, there are now 2 international studies that are going to the next stage and attempting to determine further whether or not there's a path forward here. One sponsored again by the national academies and another by the World Health Organization. And those are both slated to report out in 2020. So let me generalize a little bit more. And just for the last few minutes, I want to talk about some of the other areas of science that have posed lots of ethical and societal quandaries. In 1981, Martin Evans first isolated embryonic stem cells for mice, work for which he would later win the Nobel Prize. And in 1998, Jamie Thomson did the same for human embryonic stem cells. This was met with tremendous excitement because we now had a toolkit for repairing and regenerating every tissue in the body. And yet, as Tech Review highlighted, this is the biggest prize in biotechnology, but most companies won't enter the race. And as you know and remember, this was an enormously challenging debate of the first Bush administration. He walked this tightrope where he was able to allow a certain amount of funding for a very limited number of lines. It turns out far fewer lines than were originally promised, and this led to all kinds of calls for a reestablishment of a different proposal. And indeed, in the last 20 years, we now have wide access to embryonic stem cells. Cloning, first reported by Campbell and Willman in 1997, a startling demonstration that every cell in our body has the same genome and can actually be used to reactivate an entire organism. But this led to, again, widespread concerns that we would have armies of clones. And it was legitimate to ask, you know, in the face of lots of scientific hue and cry that cloning would be unsafe, would this ever be tried? And indeed, there were folks advocating for it. This is His Holiness Rael. And this is the well-known spiritual leader of the Raelian movement. I imagine some of you here are familiar with this, if not members. And this group was actually called to the US Congress to give testimony. I mean, that seemed crazy then, but what's going on now? I mean, it's even crazier. This actually looks normal compared to the conversations in Washington today. But there was actually fertility physicians who went forward and attempted this, and they actually published their work. So even though there was broad international condemnation of this whole strategy, it was attempted. Another application of biotechnology is the ability to use the manipulation of nuclei, in this case from an egg of a woman carrying a mitochondrial disease. Mitochondria are the small organelles in the cytoplasm that are the sort of energy factories of the cell. And they have their own DNA, and they can pass on very devastating diseases. You can actually treat this by moving the nucleus of a fertilized egg into a healthy egg. And this has been technology that has been shown to be safe in mice and monkeys. And the Human Fertilization and Embryology Authority, the oversight council in the United Kingdom for all things in vitro fertilization, as well as the US National Academy of Medicine, studied this and said it would be permissible, but there needed to be a clear course towards careful clinical trials. And yet, despite those admonitions, a practitioner in New York City moved to Mexico, where he says there are no rules, and birthed the child. Now, the post-birth analysis of the tissues of this child suggests that the child was not, in fact, cured of the mitochondrial defects. And we don't yet know what the implications of this are. But again, I think the scientific community would conclude this was a premature and irresponsible application of a biotechnology. And in the march towards ever more provocative biotechnologies, in 2017, a group from the Children's Hospital of Philadelphia, recognizing the challenge of the birth of premature babies, which are now pushing the limits of viability even before 24 weeks, actually did experiments in the premature fetal lamb, allowing that to be incubated through the point where the lungs develop, and creating the standard of ectogenesis, extrauterine gestation. And if you think about the implications of how entertaining this type of approach would introduce clear challenges to the notion of viability as a standard for abortion in the United States, this is quite an inflammatory technology. So in 2006, back in the early days of the stem cell debate, I chaired an international task force on behalf of the International Society for Stem Cell Research to come up with guidelines that would guide the conduct of human stem cell research. In 2008, we did the same for the clinical translation of stem cells. And then in 2016, we came together and updated those guidelines. What had evolved from the embryonic stem cell debates of the 2000s to the biotechnology realities of 2016 were much broader interventions with the human embryo. And so we generalized the embryonic stem cell research oversight process to include all embryo research and articulated oversight and regulatory review processes for mitochondrial replacement, gene editing, and the increasing attempts to actually regenerate embryos from component stem cells, creating embryo-like structures. And we are now just held our first steering committee. Robin Lovell Badge is going to be anchoring a update yet again to become more precise on the regulatory review of some of these emerging biotechnologies. So let me finish by saying there's a long litany that I haven't even begun to describe. There are now, we can actually make mice entirely from cells in a petri dish. We can make sperm, we can make eggs, and we can do in vitro fertilization from pluripotent stem cells. The same biology underpins the ability to make gametes in humans. As I said, scientists are getting increasingly creative at bringing together component tissues to make preformed embryos. Embryos at what we call the primitive streak stage, that first stage where the human embryo starts to take form, head, a tail, a front, a back. And this is challenging the time-honored notions of restricting embryo culture beyond that stage. Human-animal chimeras. There are legitimate interests in incubating human organs in the context of animals like pigs and the like through human-animal chimerization. But how much chimerization would we allow of the animal brain? CRISPR isn't involved in just editing cells in a petri dish. You can edit whole organisms that can be released into the environment. Mosquitoes could be engineered to be resistant to carrying the any number of pathogens, malaria, Zika, Triple E, and this is a concept of gene drives, but this would lead to an altered biosphere. The mind-machine interface, obviously creating all sorts of new issues. Bionic limbs, exoskeletons, I could go on. The fact is, what has been inspired by this, this announcement of the Chinese twins, I think, is a challenge to the time-honored tradition that began in the Silmar, that scientists could come forward and regulate themselves. There's now a call. The question is, was this a failure of scientific self-regulation? Do we need to have other interventions at a societal level, either legislative or other, to regulate the application of these technologies? And with that, I will leave you to ponder and perhaps ask me some questions. Thank you very much.
You're going to pass microphones around? Fantastic, thank you. So yes, we have some time. Anna?
Thanks for your excellent overview of the CRISPR story. So you set up the sort of main challenge in a way that 99.9% of all talk has been about, I think, which is heritable versus non-heritable modifications. But if you ask the public, that is not the distinction that they see as salient. They see the distinction as salient as between therapy and enhancement, right?
Right.
So I'm wondering, why do you think we see so little talk about that? And if I could have a separate question, it's about your view on your last question about debate and what you think an effective bioethics looks like.
So part of the reason why I think the discussion has focused on heritable as opposed to somatic is you can do somatic enhancements too, but there's lots of other ways to do that that are easier. I mean, pharmacologic enhancement is tremendously easy. So I think that the debate is sort of, it's more concerning about kind of the notion of influencing future generations. So I'll say that. On that last question, I still believe that it is part of the fundamental responsibility of the scientists to be open to independent review and critique of their work, whether it's for scientific or ethical reasons. And so, regardless of whether there will be legislation, I don't think that absolves the individual scientific communities from having the responsibility of going through institutional review, going through peer review. And so we may layer on other types of regulation, but I think we'll still be getting together and debating it within our own communities.
So I'm going to ask a question real fast. So I know there are lots of hands out there. Do you mind going back to your slide with the green and red stop and go lights?
Sure. Here, let me see if I can.
Right. So that's, I mean, it's a lovely slide and it's very helpful and reasonable. And I'm sort of sitting here thinking about it, asking myself the question of what other work would one need to do in the space. Generally, I realize that part of the reason it seems so reasonable and sensible is precisely because it's inside a medical frame. It's inside the professional commitment to disease prevention and so forth. And so I think my question is really about adversarial actors of various kinds and how the scientific community thinks about that. So I think one of the challenges thrown up by these technologies are just that you started with Orwell. So how do state actors, how does China think about something like this and so forth? And where is that conversational space?
I can tell you that the organizing committee in Hong Kong spoke quite a bit about why the first report occurred in China. And there was lots of speculation, but I think one of the prominent issues was the concern about how a government that could support a one-child policy that already had sort of an intellectual, cultural tradition of manipulating the sort of the human population. I think that's as far as I was really willing to explore the issue. I think it does create a quandary about how state actors would use this. And the government has changed and evolved dramatically in China, and so I don't want to put too much emphasis on China alone. We have seen other communities come forth in Russia and other around genome editing, and so I think there's a period during which there's enough of a sense of going rogue, being the intrepid pathbreaker that is driving some scientists to do this.
Thank you. I saw lots of hands against Naomi.
Thank you very much for the very compelling lecture. So I would like to ask you about your responsibility and self-regulation framework and offer you a dichotomy of how to interpret it. So on the very cynical view, scientists are really worried that the public is going to have a quite critical pushback and not let them do the productive aspect of this research. So, for example, GMOs come to mind, vaccine kind of reactions, or the other kinds of reactions that you listed in terms of stem cell reactions, for example. And so therefore, we need to make these calls to regulate ourselves and not be subject to what the public wants of us. But the forgiving end of that is that no, we truly are very worried about what might happen, but we also don't know really how to respond in these situations with these ethical considerations, and therefore we truly want the public to weigh in and have input. I'm wondering where you see yourself on this dichotomy, and also feel free to push back against it if you disagree with that. And if it's the forgiving view, how do you truly want the public to be involved in these conversations?
Yeah, I understand the potential criticism that self-regulation is essentially advocacy masquerading as inquiry, right? I mean, we somehow know better. I'm guilty as charged to some degree. I think if, from the scientific point of view, I think the GMO experience has sort of chastened a lot of scientists that we assumed too much. We assumed too much acceptance and receptivity to something that the scientific community thought would be a boon to humanity, the food supply and the like. And I think there is a concern that we not let misinformation or disinformation stand in the way of what we think are potentially very beneficial applications of this technology. I think that's even more compelling when you talk about gene drive. Gene drive is very hotly debated. So there is technology where we could modify mosquitoes to be resistant to malaria and certain other vector-borne, make the vector resistant. And those aren't going forward in part because of a tremendous insecurity because of what we don't know. But lots of medical technology has gone forward without fully understanding the risks. And the risks of malaria are real and quantifiable. And so you're trying to take a quantifiable, devastating risk and weigh it against an unquantifiable, unknown risk. That's a challenge. And I would argue scientists shouldn't be alone in making that balance. I think scientists should be at the table informing that debate, but there are many other considerations that factor in. I hope that addresses your question.
Just one short note about China. I think it's notable that two of the leading bioethicists in China, Rentong Chu and Zhai Xiaomei, have published some of the most outspoken critiques. I mean, going far beyond what many of us in the West would say, and nothing's bad happened to them. So there seems to be some space in they're in Beijing, and so there's space there for that point of view. It's not in lockstep. That's important to.
And they spoke, they were both, Jiankui was part of the organizing committee, and the other gentleman was at the
Chu,
meeting. Yeah. And they spoke very aggressively at the meeting.
Yeah.
Right there.
So, but my question is somewhat different. We can talk about national world governance, this sort of thing, but how about here at Harvard? Right now, we have an escrow.
Yes.
The escrow is a
Say what the acronym is.
yes, it's an embryonic stem cell research oversight committee. So this thing is, and I'm a member of it, so I'm speaking here from the inside, I guess. It's a committee of people from law school, all over the place, with scientists, of course. It's totally voluntary. People, mostly stem cell scientists, bring their protocols and say, take a look, what do you think? And what happens, there is a vote. I'm not sure what would happen if there were an absolute no vote, but what actually goes on almost all the time is the committee gets informed by the scientists doing the work, and that kind of education is always needed because the scientists doing the work know more about it than anyone. And then there's a conversation, and from this comes a meeting of the minds in almost all cases. Now, it's those of us who are on the escrow are not represented. We weren't elected. We're not. Nobody gave us the authority to speak for the public and so on. But it has sort of worked as a model for creating an on-the-spot forum for discussion among people of diverse points of view. Only rarely has there been a difference that seemed unbridgeable. And I think the scientists have been comfortable with this. So far, the campus seems to be. What do you think this is a kind of model? First of all, do you think that this is working at Harvard? And secondly, can we draw any lessons from that to the broader sphere?
Well, let me try to make my point by highlighting a counterexample. Jiankui, the Chinese scientists, claim to have gone through an internal review process, an IRB review process. It wasn't an escrow process, it wasn't an embryo process, it wasn't an embryo research, but it claimed to have had scientific and institutional review. And they supposedly approved it. And I have seen many other protocols at other institutions for various stem cell interventions be approved through an institutional review process that just, I'm convinced, or it was an inadequate review. Your process and your conclusion is only as good as your process. And we know it's a flawed process. I think it's better than no process. And I think we aspire to continue to be more thoughtful. And part of the reason that we're organizing a revision and update of the guidelines is to be more prescriptive. We were asked by lots of escrows to clarify uncertainties about how iPS cells are treated, things of the like. So I think we're constantly moving towards a better and better process, but we're only approximating what we aspire to. We know it's a flawed process, but it's like many things, democracy and the like, it may be better than most other processes. I'm eager to hear other ideas if we have a better one. Yeah.
So there were some student hands towards the back. I don't know your names.
As we already know, CRISPR in itself is like a scientific breakthrough in the science community, and it's good for like curing diseases and to prevent different diseases as well. And I know that morally and like in terms of the press, it could be like really like unbeneficial because you could use it to like make like certain babies and embryos like stronger, and that could be concerning like morally. But I wanted to know, even though the CRISPR in itself, the technology isn't 100% accurate, when will the next humans be tested?
Well, the point is that I think you're implying, and is endorsed by all of these international bodies, more research is needed. but that research would be on embryos in a dish, not embryos that would be brought to term, not embryos that would make a baby. And all of the groups that have looked at this, those groups that at least support embryo research, many groups don't because of opposition to the notion that an embryo is a person, but they all call for more experiments before we got to a clinical trial. And those experiments should include animal experiments, so we need to do more mice and understand the implications in mice. We can do it in monkeys before we subject another child to the same kind of experiment. It's a very good question.
Up front.
Eric, yeah.
I'm not at all over here. I can't see you guys well. The lighting is such a, you could actually put your hand up. Okay, so it's
Just to be provocative, and assume for the moment safety, efficacy, all that stuff. In this slide here is pretty much like from a doctor-patient perspective, and nowhere is there kind of a talk of your talk is the cost to society of treating these diseases once they're there, and so obesity, diabetes, all sorts of things are incredibly expensive for society, and essentially it's one body than populous one. So, assuming that it all worked and all that stuff, is that something that should be a factor of consideration, or is medical ethics that it's only doctor patient doesn't have the cost to society?
I think in the discussions that I've had of this sort, what I've taken off actually from the slide, just in the interest of time, the alternatives are somatic therapies. And those have to be weighed against the germline intervention. And arguments have been made, even if we come up with a reasonable somatic gene therapy for sickle cell anemia, doesn't it make sense to correct it in the embryo? And you don't have to do it again and again in every generation. That's an argument that is made. Would there be any cost to eradicating the Huntington's disease from human gene pools? I find very few people would argue that it would come from. So you do think about weighing the cost, the actual cost, the feasibility, utility. I think those are very realistic.
right. Well, I was just say I think the provocation was the notion that there might be enhancements that, from a medical point of view, would be negative, but from a social point of view, if you're thinking about public investments in particular health problems and obesity and things.
Well,
anyway, just
that is provocative. That is much more provocative. I'm the dean of Harvard Medical School. I have to be careful what I say in public, so I didn't take the bait.
Right, fair enough. So but it outlines the way in which there's a set of ethical considerations inside medicine, and then the question is like, what are the other domains of inquiry that are affected by changing medical capacity, right? And then what's the ethical framework for engagement with those domains?
Well, this is not an area where I have great expertise, but I have heard very compelling discussions about eugenics and the tacit justification saying, well, eugenics is okay if we knew how to do it. And that starts to add all sorts of layers to the concerns about the social applications of how this would be used. Some scientists have said if we could figure out how to make someone learn better and engineer, wouldn't we want to engineer the entire population to have that? Well, that's a fanciful experiment, because the real issue is if it were to be useful, who would get to use it? And how would it be distributed? Could we ensure that there would be broad and universal social access? That's where it starts to get a little more challenging.
How Collins will.
Should I use the mic or?
Well, you doesn't sound like you need a mic.
No. I don't. So as somewhat of a happy counterexample, in vitro fertilization was a radically risky technology until Louise Brown was born in 1978. at which point essentially the ethical concerns, while they kind of bubble to the surface around then, just kind of rapidly evaporated to the point where it's, you know, widespread adopted technology.
Yep.
Not only here in the United States, but elsewhere. What makes IVF, what distinguishes, or at least in your opinion, the public's ethical perception of IVF, which essentially is, I don't even think people think about it anymore, to this, right, to CRISPR? It seems like the power to create a child where one would not have otherwise been created is much more powerful technology than tailoring one.
Yeah. So let me say this. IVF was introduced after decades of embryo experimentation. And even though, you know, Steptoe and Edwards and all were very intrepid in doing the actual experiments, they were reasonably confident, based on lots of animal experiments, they could do it safely. But I would argue with your notion of whether it's still broadly accepted. There are still widespread concerns about IVF and sex selection. American Society of Reproductive Medicine basically doesn't allow it except in the cases of family balancing. If a family has had 4 males and they want a girl, they'll do sex selection or something like that. But there's a lot of controversy around sex selection. Also, when you start talking to folks about whether they should use IVF and other kinds of embryo selection, it becomes controversial. So I think that this is, you know, this is the framework that I think is at least one that anchors us on the sort of disease model versus the kind of enhancement model, and that's where I imagine people are going to find the left side of this more like IVF and the right side of this more like sex selection.
Come up here, and I'll come up here. Wrong way, sorry about that. Go for it. I'll back over here.
Hi, Dr. Daley. Thank you so much. I think one of the things that most comforts me about this is that you.
Something comforting?
Yeah. Yes. No. I mean that there really. I have been wondering for several years now what the scientific community is talking about when I read all these CRISPR articles, and I'm considering the implications of all of this. And so it is actually very comforting to hear that you've had a number of summits and that the experts are putting their brains together, and that people are very concerned about this. And I actually very much appreciate you addressing all of these things. I am kind of an interesting amalgamation of population interests that you have here. I'm a member of the community here. I'm not a PhD. I am a member with a chronic disease illness that could potentially be affected by these technologies, and so I maintain a very avid interest in how they can be useful. I am also a military wife, and I think there's several things that haven't been brought up in the talk that I'm very curious about. So one word that was not used at all was warfare, and I think there was an implication of that with the state actors, but you know, the U.S. Army is looking at the future of warfare, and yeah, absolutely that, and also
Exoskeletons.
how to remove humans from the equation. So what is the definition of a human, and how can that be altered by CRISPR? Then another thing that was not talked about in terms of costs was, let's take a look at the HeLa example, where people whose tissues are used in scientific research are currently not compensated for that. You can create an entire company off of those cells that can make millions of dollars, but the people who donated or whose tissues were used, sometimes without their knowledge, are not compensated. And so I think there's a real concern ethically, especially when you're modifying DNA and putting cells, what is the transparency back to those patients, to those donors, and how do they get compensated? Because is it ethical to continue without compensating? So I think that's a big question.
That's a great set of questions. If you don't mind, we'll let you.
Well, and actually this is not so much a question, but just you made one other huge point about alternatives, and the alternatives with somatic things on disease prevention. I do think IVF is still very controversial. I think it should be controversial. And I think one of those alternatives we still have to push as a society is adoption, looking at people in society raising children in a very different way rather than spending billions of dollars on designing the perfect child. So I don't know if there's necessarily an easy question to respond to, but it's more of a framework of there's a lot more to consider. And to answer your last question, I do believe that some kind of regulation is necessary because sometimes some scientists, as we've seen, just can't contain themselves, and understandably, but that's really the part where I'm curious as to your thoughts on that. Thank you so much.
Well, thank you. I'm glad that you raised some alternative. You're establishing an alternative voice, which is part of why we want to have more open debate. Fortunately, I think on the compensation and use of tissues, that has come to the fore quite dramatically. I think it's been popularized by the book, and now it's a broad international standard that in a consent for obtaining any tissue, the whole notion of whether there's value present in the future, and whether there's compensation present or future, is part of the standard of the way we do informed consent nowadays. And I would just say the last thing, most of the scientific conferences that we go to, most of the conversations you wouldn't find very interesting. This one is an unusual thing. That's it, yeah.
I just wanted to go back to Jacob's question, and just to clarify, my understanding was that what makes IVF different from CRISPR babies from a kind of ethical standpoint is that you're actually changing the hereditary line with CRISPR babies, which is why kind of historical debates about eugenics and things like that are useful in a way that it raises a whole set of questions that are different.
Yep. Mm-hmm. I mean, IVF is, it's still random. It's mating in a dish, if you will. And there are lots of arguments that, given that sex is cheaper and more fun generally, that it's unlikely to give way to in vitro sex all the time. Making the point that anybody. Actually, I'm being flip, but I just heard Alton Charo make an argument that is attempting to sort of back us off the worries about the applications of this, and it has to do with the opportunity that has been presented to couples wanting to have children. Even given the opportunity to go through massive notebooks giving all the details of the background and the SAT scores of sperm and egg donors, most people actually don't avail themselves of those properties. Most are trying to find donors that have similar properties to themselves, similar traits, similar appearance, similar ethnicity, similar heights. Not everybody is going for the 6'4", 800 SAT type of thing. And she argues, she says, I'm not sure that we should be worried so much about these kinds of selections, because even in the face of technology that could allow us to do more directed mating, people don't elect it. And I thought it was an interesting perspective.
So I'm afraid we have to call a halt for this evening, but thank you. You guys are incredibly wonderful. Is next Friday. Somebody knows the answer to that. October 5th, Teresa Bijan from Oxford. And then after that, November 7th, our first civil disagreement event where we will have two speakers about immigration on either side of the political spectrum in a moderated conversation. I will moderate. We'll have fun. Anyway, so come back again. Thanks for joining us.